Alright, I'm going. Camera angles still aren't sorted out, but let's do this. Biological molecules. First topic in biology. Probably one of the easiest ones to ease you in with. It's quite a nice topic to get your head round. So let's begin. First thing I want you to do, imagine an animal. It could be a dinosaur, it could be a worm, it could be a chicken. Basically anything that you want, any organism that you want. And now we take it and we zoom in on those creatures. Zoom in a bit further and you see all of the organelles inside of these cells. Zoom in a bit further. No, not that far. We're not chemists. Pull it back out. Right there. Molecules. On the molecular level, you see there's hardly any difference between the two substances. Pretty much all organisms we know of have simple fundamental molecules that make them up. These are called monomers, single sections of a longer chain. I'm sure you've heard the term polymers before. Usually we talk about plastics when we're talking about polymers, but in the case of science, whenever we talk about polymers, we refer to any long chain of substance that is made up of individual base units called monomers. Monomers are a lot smaller and will connect to a lot more monomers to make a polymer. It's not usually just one or two connected to each other. And we have a lot of examples of polymers. You can think proteins, starch. It's just so easy for the structures to get massive for a molecular scale when you're connecting base units together. So as far as monomers go, the AQA exam board lists 3 on their exam specification. So you've got nucleotides, amino acids and monosaccharides. But there are plenty of monomers out there in science and the real world. Think ethylene, and that's pretty much all I can think of off the top of my head. So how do we make these long polymer chains? Well, there are 2 reactions that we have to consider, and the first one being condensation. This reaction occurs in the presence of certain enzymes, depending on which molecule is at hand. But effectively what happens is, at the end of each of these molecules will be at least 1 oxygen and 2 hydrogens in each scenario, which I'm sure, as you're aware, would create a H2O molecule, causing more water to be on the product side than is on the reactant side, hence the term condensation. This reaction can be done many times to create a long-stranded polymer chain. In some situations, however, we have to break down those polymers back into their constituent molecules. Examples are things like digestion, where very long protein chains won't be able to fit through cell membranes to get to your cells. However, we can break up these molecules using a reaction called hydrolysis. Hydrolysis simply means the breaking of a substance using water. And in the case of polymers, this is no different. Using certain hydrolase enzymes, we can effectively cut the molecules. It's different in which polymer that you're referring to, but we'll get onto each individual polymer in a separate video. So at the end of that reaction, you end up using the water that you have initially formed through your condensation reactions, and it can be reused to split these polymers back into monomer structures.